Hi everyone, welcome to my presentation on evolution of respiratory adaptations from aquatic origins to terrestrial life. I'm David Hill. Today's presentation will cover the evolution of respiratory systems as vertebrates moved from water to land, a transition that required profound physiological and anatomical changes. Our objectives are fourfold. First, I'll review major changes in gas exchange systems during this transition. Second, I'll discuss the evolution of respiratory adaptations across several key clades. Then, the evolution of fully terrestrial tetrapods, amphibians, and amniotes. And finally, I'll touch on specific reptile groups. Lastly, I will talk about two animals I picked out. In this slide, I want to provide a detailed overview of the fundamental differences between aquatic and terrestrial respiratory systems and the challenges vertebrates face when moving from water to land. Let's start with aquatic respiration. In water, vertebrates primarily use gills to extract oxygen. Gills are very effective in an aquatic environment because water contains a high concentration of dissolved oxygen. And gills are structured with a large surface area, allowing for continuous water flow and efficient oxygen uptake. The water that passes over the delicate gill filaments allows oxygen to diffuse directly into the bloodstream. However, when vertebrates made the transition to land, they encountered a set of entirely new challenges. First, air has a much lower concentration of oxygen than water does. More importantly, air is dried compared to water, and the tissues of early vertebrates were not adapted to prevent water loss. This desiccation can severely impair cell function and overall metabolism. Additionally, terrestrial environments bring with them wide fluctuations in temperature and humidity, which further stresses the respiratory system. These challenges necessitated the evolution of a completely different respiratory structure, the lung. Unlike gills, lungs are internal sac-like organs that are capable of retaining moisture while allowing for efficient gas exchange. The walls of the lungs are moist, which facilitates the diffusion of oxygen from the air into the blood, while at the same time minimizing water loss. However, lungs are not just a simple swap from the gills. They required significant structural and functional innovations. For example, early lungs had to evolve from a primitive respiratory pharynx, gradually becoming more compartmentalized to increase the surface area for gas exchange. At the same time, the circulatory system had to adapt to support this new mode of respiration by efficiently transporting oxygen from the lungs. The first major clade I will discuss is represented by early fish groups known as Porolipiformes, Osteolipiformes, and Elpistoscidigids. These animals exhibit some of the earliest signs of lung evolution. Although still primarily aquatic, they developed primitive sac-like lungs derived from the respiratory pharynx. These structures were not highly complex, but they represented an important evolutionary experiment that allowed these animals to exploit shallow water habitats where oxygen could change. Additionally, these adaptations may have provided a survival advantage in environments where predation was high. This is spoken about by Lung and Carrier et al. That found forcing early vertebrates to explore new ecological niches were driven by these factors. Next on our evolutionary timeline is Tiktaalik. Tiktaalik occupies an important position on the evolutionary timeline because it shows a mosaic of features that are characteristics of both fish and early tetrapods. First, consider the rib cage of Tiktaalik. Unlike typical fish whose rib structures are adapted solely for buoyancy and streamlined swimming, Tiktaalik ribs are robust and flattened. This suggests that its rib cage is beginning to support its body weight in an environment where gravity plays a more critical role, a necessary adaptation for any animal venturing onto land. These robust ribs would have provided structural support to protect the developing lung area and to resist collapse under the force of gravity. Daischler and his studies show us the pectoral fins of Tiktaalik are particularly revealing. Detailed morphometric analysis demonstrated that these fins contain elements that are homologous to the limb bones of tetrapods. Specifically, Tiktaalik's pectoral fins include parts that resemble a humerus, the radius, and the ulna. Although these structures are not fully formed limbs, their presence indicates the evolutionary groundwork was being laid for the development of weight-bearing limbs capable of supporting locomotion on land. Furthermore, Tiktaalik retains some of its aquatic traits, such as parts of the gill structure, while simultaneously showing emerging characteristics that would later support lung-based respiration. This blend of features is exactly what one would expect from a transitional form, a creature in the midst of adapting to a new terrestrial way of life. As vertebrates continued to transition onto land, reptilomorphs emerged as a critical group, bridging amphibians, and amniotes. In these transitional forms, we see further refinements in lung structure, with lungs becoming more efficient and better suited for continuous airflow. This evolution meant that these animals began to rely less on cutaneous respiration, a system that is effective in moist environments, and more on pulmonary ventilation, more effective on land. These changes were driven by the need for oxygen uptake to support increased metabolic rates in a fully terrestrial environment. The respiratory innovations observed in reptilomorphs laid the groundwork for the development of the complex lungs found in amniotes. Modern amphibians provide an excellent example of how early respiratory adaptations have been retained and modified. Amphibians, such as frogs, still rely on simple sac-like lungs ventilated by buccal pumping. Additionally, they depend on heavily cutaneous respiration, absorbing oxygen directly through their skin. The respiratory strategies are well suited to their life history traits, which include metamorphosis from aquatic larvae to terrestrial adults, and a strong dependence on moist, humid environments for survival and reproduction. Dater explains that this dual system ensures that even when conditions change, like variation in moisture or oxygen, amphibians can maintain proper oxygen and support their physiological needs. As vertebrates became fully terrestrial, amniotes evolved complex lungs that featured internal subdivisions, alveoli. This greatly increases the surface area available for gas exchange. This advancement was essential for supporting higher metabolic rates and more active lifestyles. In addition, many reptile groups such as Testudians, squamates, and serpentines further specialized their respiratory systems to minimize water loss and facilitate efficient thermoregulation. The evolution of the amniotic egg in these groups also reduced their dependence on water for reproduction, allowing them to colonize a wider range of terrestrial habitats, as shown by Goris et al. In 2000. Now, let's examine a case study that compares the respiratory adaptations of two groups, frogs and snakes. This group illustrates how evolutionary pressures have led to distinct strategies for oxygen uptake in vertebrates. Starting with frogs, these amphibians possess relatively simple sac-like lungs that are ventilated by a process called buccal pumping. In this mechanism, a frog lowers the floor of its mouth to create negative pressure, drawing air into its lungs, and then raises the floor to force it out. The active process is critical for lung ventilation. However, frogs don't rely solely on their lungs. They also perform cutaneous respiration, meaning they absorb oxygen through their moist skin. This dual respiratory mode is particularly advantageous in the moist environments that frogs inhabit, where water is abundant and skin remains moist. Such an arrangement is closely linked to their life, as frogs undergo metamorphosis, transitioning from their aquatic gill-bearing tadpoles to terrestrial air-breathing adults. In contrast, snakes, which are representative of the amniotes, have evolved a very different respiratory system. Most snakes possess a single elongated functional lung that is highly complex and subdivided into alveoli. These alveolar structures provide a large surface area for cutaneous and efficient gas exchange. Because snakes generally inhabit drier and more thermally variable environments than frogs, this lung architecture is adapted to maximize oxygen uptake while minimizing water loss, a critical adaptation for survival in arid conditions. By comparing frogs and snakes, we see a clear divergence driven by their environments. Frogs maintain a flexible dual respiratory system that works well in moist habitats, while snakes have evolved a more efficient single lung that conserves water and optimizes oxygen extraction in drier settings. Environmental pressures have been the primary catalyst for the evolution of vertebrates' respiratory systems. As vertebrates transition from water to land, they encountered a dramatically different environment. Oxygen is less abundant than in water, and the risk of water loss increases substantially. In addition, terrestrial habitats are characterized by significant temperature fluctuations. These challenges forced a complete overhaul of the respiratory strategies. Initially, they relied on gills, which is efficient in water, but as they moved to land, vertebrates evolved lung-based systems. In amniotes, lungs became highly subdivided, with numerous alveoli dramatically increasing the surface of available for gas exchange while conserving water. I selected peer-reviewed articles that provide complete details, each including an abstract, introduction, methods, results, and discussions. Thank you so much for attending.